Wonderful to have you visit with us, Erik. Thank you so much. A lot of the fellows are always interested in what your experience was. How did you hear about the foundation? What do you remember about the somewhat famous interview?
So I did my postdoctoral fellowship with Leonard Mookie at the Gladstone Institutes in San Francisco. And there had been prior Giannini fellows there. And so I became aware of it through the institution. And was eager to apply. I had been a neurology resident at UCSF and was just getting back into the lab after, after 6 years of being in medical school and residency, trying to catch up on all the new techniques that had been invented. Science moves pretty fast. It doesn't, you don't always realize it until you step away for 6 years to do clinical training. And was getting started, had identified my project and gotten a little bit of preliminary data and put the application together. Was the first grant that I wrote, and remember being excited to get the invitation to go down to Pebble Beach, and because I had not I'd lived in the Bay Area for 4 years by then, but having spent most of that as a neurology resident, didn't get out and do much traveling around. So I hadn't seen much. I hadn't been down there. I was excited to kind of check it out. And then I remember getting the roster of who was going to be on the committee, and building, kind of looking everybody up, and oh my goodness, it's all chairs and deans from all over the state, and trying to figure out who studied what, so who would know what, and kind of what kind of questions I might get, or how to tailor the presentation. And I think that's also it's great experience to be able to channel your science to an expert audience, but not an expert in your field. Some people who understand the experimental design and the kinds of approaches, but not necessarily the background and the rationale for the question. One of the early lessons in being able to really explain why what you want to do is important, and certainly is a critical part of writing a grant that I learned there. Yeah, and I remember I'm walking in and seeing a lot of other Giannini Fellow candidates waiting in the lobby to go in, and have a chance to go in and present. It was an exciting day.
You completed your residency in neurology at the University of California, San Francisco, where you served as chief resident in neurology. You then completed a clinical fellowship in behavioral neurology at UCSF and resumed basic research at the Gladstone Institute of Neurological Disease. You joined the faculty at the University of Alabama at Birmingham in 2008. So now we went from 2004 to 2008. That was a lot of stuff that happened once you got that fellowship, and then when you went to the University of Alabama. Talk to us a little bit about that.
I had a really neat opportunity to sort of custom build a fellowship. When I finished my residency, I knew kind of what I wanted to do. I had gotten my Ph.D. in neuroscience, studying basic mechanisms of learning and memory in brain slices. And so I was interested in memory, and it was a fairly natural progression to go from having a basic science interest in memory to then becoming a neurologist. That Alzheimer's disease would be the area that I would focus my career in. But then the question comes up: how much clinical fellowship training would I need, and how to balance the need for clinical fellowship training versus just what's critical, getting back in the lab and starting to be productive in the lab, because that was really where I wanted to primarily focus and build my career. And in the end, I decided not to do sort of a full-time, taking another year or two of full-time clinical fellowship training, but rather was able to sort of build that into a basic science fellowship. So I was really from the July 1st, the day after I finished my residency, was in the lab with Dr. Mookie at Gladstone, but also then taking basically a half a day a week to sometimes a day a week to be in the clinic at the Memory and Aging Center at UCSF with Bruce Miller, and starting to build up clinical training. So I sort of integrated my clinical fellowship training and I'm now considered a fellowship trained behavioral neurologist, but I did that without having, the full-time 1 year clinical fellowship training, kind of integrated that over a little bit longer period while I was in the lab. I think that really worked out well for me. As an MD PhD, you've already kind of invested a lot of years along the way, and you get kind of cautious about having too many more years go by that before you really kind of get into the key productivity years of what you really want to be doing. So that was a good way for me to shave that down a little bit. And it was really fortunate to be at UCSF, where we had both excellent opportunities for clinical training and excellent opportunities for basic research in neurodegeneration. And really importantly, to have mentors, Bruce Miller and Leonard Mookie, who I knew that they knew each other and got along with each other. And I think the risk when you have a dual mentorship arrangement is if your two mentors aren't on the same page and they kind of pull you in different directions, you're kind of on the rack. And that's not where you want to be. You want to make sure that it's an arrangement where the two mentors are going to both see eye to eye on what you should be doing and how you're spending your time and effort and work together with that. And that was totally the case with me. And so that worked out really well. So I was able to really kind of get back in and establish a track record of being productive back in the lab while still keeping my clinical skills sharp, and adding to them with the behavioral neurology training, and really to kind of get essentially from the day I finished residency into the balance that I wanted to have, which is a sort of half day a week clinical practice with the rest of the time devoted to the lab and basic science.
What was it about Alzheimer's that caught your attention, that you thought this was something I want to focus my career on?
I think primarily, it was that I was well versed and had a strong interest and background by that point in mechanisms of learning and memory, and having them become a neurologist, you need to kind of you focus on the diseases and not on how it's supposed to work anymore. And so Alzheimer's disease was just naturally it's the most common disorder of memory. And so it was a fairly natural fit. Of course, it's a big problem. I don't think we all wasn't getting quite as much attention, and certainly not as much funding as it is now at that point, but it was certainly recognized to be a major problem and an important one to address. I actually, and I think the other thing was that it was becoming, earlier, like when I was in medical school, I considered actually whether to do neurology or even to do psychiatry when I was finishing up as a fourth year medical student. Because, a lot of the psychiatric diseases are really quite interesting as well. And in a way, almost to some extent, more so to me at that point. Because my thing at that time was, that the neurodegenerative diseases were just diseases of the cells dying, and really all you would really be studying is mechanisms of cell death. Whereas the psychiatric disorders were more disorders of neuronal dysfunction, where you really were kind of getting into the function of the brain and how that was going awry. Of course, when I realized that there's a whole lot of dysfunction that's going on in Alzheimer's disease before brain cells die, and that's really most of what my lab has ended up studying. Where we don't so much focus on the end stage formation of a neurofibrillary tangle and the death of the neurons. We're more interested in that kind of the neuroscience interface of the cascades of signal transduction that are starting to go awry and leading to neuronal dysfunction before there is neuronal death. And once I realized that you could kind of do that it wasn't all just studying death pathways and why a cell died, that there was actually dysfunction before that was kind of what sealed the deal for me.
Give us a sense of context. What was known in 2004 and what has changed about our knowledge today?
It's a great question. I remember as a medical student, I believe it was as a medical student when the initial discoveries were made of what enzymes were the ones that were responsible for producing amyloid beta, the kind of the key toxic peptide that's believed to be a main driver of disease that the genetics have pointed to for a long time. And when we figured out that base 1 and presenilin were the two enzymes that made the two cuts that generated amyloid beta from its precursor protein, I remember people telling me, like, you know, I mean, Alzheimer's disease, it's kind of we know what we need to do. We just need to develop inhibitors of these two enzymes. You'll slow the production of this amyloid beta, and you know that's going to be the treatment for Alzheimer's disease. And by the time you get finished with your medical school and residency, I mean, this might not even be a disease left to, I mean, not that much left to do at that point. Of course, you know, 20 years later, here I am, and there's obviously still plenty to do. And just, you know, I guess the lesson learned from that was that, you know, we never know as much as we think we know about the way things work. In 2004, when I was starting my fellowship, was kind of the key questions were, it was just sort of coming to the end of the period of the war between what was called the Baptists and the Taoists. Those who believed that amyloid beta, which makes the amyloid plaques in Alzheimer's disease, was the main culprit and the main driver of disease. And people who thought that tau protein, which is the main component of the neurofibrillary tangles in the disease, was the main culprit and the main driver of the disease. And there had been a period where, you know, there were these kind of big fights at meetings between people who studied amyloid beta and the people who studied tau about which one was more important. And the project that I ended up being the kind of the focal point of my postdoc in Leonard's lab kind of got at that. We didn't really set out to address that question, but we did an experiment asking the question of how much tau contributes to deficits that are caused by amyloid in a mouse model. So we took transgenic mice that were overexpressing amyloid precursor protein, the kind of the standard Alzheimer's disease mouse model that overproduces amyloid beta and develops plaques and develops memory deficits. And we reduced the expression of tau protein, knocked it out of those mice, or knocked it down by 50%. And we're somewhat surprised to find that that had a strong protective effect. So these deficits driven by this mouse model that's amyloid dependent were also affected by reducing tau. And that was kind of, we published that in Science, and that was really the key paper of my postdoc that, you know, was kind of the linchpin that helped me get a faculty job and became the foundation of my first R01. And you know, the story that we could kind of tell around that was, it helped to kind of end that Baptist versus Taoist war, because the answer was it was both, and they worked together, and tau was even important in the problems caused by A beta. Yeah, so I mean, that was kind of the context of where the field was at the time that I was just getting started in this.
Does Alzheimer's begin at a moment and it's like the switch is turned on, or is it little steps that lead to something that someone says, there's a problem here?
From a symptomatic point of view, it's an insidious disease that you're never able to pinpoint a precise onset. And we try to talk clinically with the patients and their families. They're trying to understand what that first symptom was, and really, you know, the first time that there was really a significant concern about their memory. But it's completely insidious and difficult to pinpoint. Now, biochemically, molecularly in the brain, there probably are multiple pathways by which that can start. There are different genetic risk factors. There are possibly some different environmental influences, you know, possibly a role for head trauma in different, you know, different things like that. So different people may come to it by different ways, and some of those may be a little bit more discrete than others. But I think that, in the bottom line, we don't yet still fully understand, for the vast majority of people, why they're getting the disease and what the initial, you know, what those initial causes are, what those initial steps are. I think the genetics, because they have a causal component to them, because they imply that genetic change is actually that is driving the risk are best clues to that, and that's kind of why, as my lab has grown and evolved, that's sort of been the organizing principle, is trying to better understand taking the findings from GWAS studies that have been unearthing, tens of different genetic risk factors, most of whom it's not apparent at all how they would be contributing to the disease, what they're even do in the brain. Trying to understand that and what that might tell us about the initial steps in causing the disease.
You go to your doctor and you get a blood test and you check your heart and your blood pressure, et cetera, and there are just signs there if there's something going on. With Alzheimer's, it could be years where there are no signs that are revealed in the normal clinical process. Is that correct?
Yeah, that certainly is correct. The things that are starting to change that, when I did my training 15, 20 years ago, even up until the diagnostic criteria published in 2011 were still based primarily on just the Alzheimer's disease, based on the presence of a dementia syndrome, some cognitive impairment that has been progressing gradually from an insidious onset. That's really all you kind of needed to make a diagnosis of probable Alzheimer's disease. One of the big advances was to be able to do molecular PET imaging, for the amyloid plaques, and then which previously, we didn't know if somebody had plaques unless you looked at their brain pathologically. Those studies revealed that the plaques start forming 15 years before the first symptoms of the disease. So that was kind of one of the first pieces of evidence, kind of for what you're saying, that some objective clinical biomarker type evidence that the disease process has begun, even in someone who doesn't have any symptoms, but certainly at the time of their first symptom. Of course, a PET scan is very expensive. It's gotten FDA approved, but has still not been able to obtain widespread coverage by insurance, and so it's almost never really done in a clinical setting. I think the next step, and the big hope over the last year or two, is for blood tests. So there are now very sensitive blood tests that detect abnormalities in amyloid beta protein levels in the blood, and that is looking like it correlates quite well with a positive PET scan. And that will be, as the technology gets modified and rolled out, that will become, I think, probably something that can be done cost effectively on a wider scale. And that probably, over the next several years will become a path to earlier diagnosis.
So what have you been able to do to translate your clinical care into actual patient care and your research? Have you been able to connect those two things?
Yeah, that's a great question. When I did the MD PhD, people would ask me for a long time, "Are you glad you did MD PhD?" And I always would punt on the question. I'd say, I can't really answer that question until I've really been doing my job for a while, and I can tell you whether I really use both sides of it. Because in the end, if I don't end up using both sides, if I end up just doing science in the lab with my PhD and never see patients, or only see patients and don't have a lab that's doing research that needs a PhD, then I don't know if I would say that I was really glad that I invested all those extra years and hard work in doing the MD PhD. I think now that I've gotten to the point where I'm doing both, I can definitely say I'm glad I did both, and that it's been a good pathway. It took a while to adapt to those transitions. It's always hard to go, from being in the lab to being in the clinic. It's a very different environment, a very different mindset, a very different approach to everything. But it got much easier when it wasn't, being an MD PhD student where you go 2 years of being a full-time medical school to being, 4 years of being a full-time graduate student, and then back to being, these week-to-week changes where, up until, Monday and Tuesday I'm in the lab, and then Wednesday afternoon I go to the clinic, and then Thursday I'm back in the lab. Then the rhythm gets easier to manage. And for me, the key, I guess this comes back to the question that you asked earlier about why choosing Alzheimer's disease. Behavioral neurology and Alzheimer's disease is an area that is really amenable to doing this. The clinical demands are not, that extensive. If you think about, at the other end of the spectrum, some kind of very complex surgical subspecialty that just requires a great deal of manual dexterity, and you don't want somebody doing that operation who only does it once a month. You want somebody who kind of does it every day, and that, or even just a setting that's more acute care, where you need to see patients on a daily basis. This is a chronic condition. Generally, it's obviously has a huge, it's a fatal condition with high morbidity, but it's not generally, there are not a lot of acute care issues. And so you can kind of show up to clinic half day a week and be fully in the clinic, and then, not have it detract from a research career the rest of the week in a large way. And then I guess I've also tried to develop the questions that we're asking in the lab in a way that will really help facilitate that translation. At the beginning, of course, when really just getting the lab started, I still found it beneficial. I think my patients enjoyed the fact that I could, in the course of the visit, talk about what was the latest in Alzheimer's disease research. That I kind of had that research side of it. But I also think that being really well versed in the clinical disease and what the real current pressing issues at the forefront of the clinical care of that disease help to inform the research. And one example of that, I think, is a project that we had on frontotemporal dementia. So my lab works on both Alzheimer's disease and frontotemporal dementia, which is kind of FTD. Frontotemporal dementia is one of the other common, nothing is as common as Alzheimer's, but it's in that second tier of common neurodegenerative dementias. But it has a very different symptom profile. It's not primarily a disorder of memory and cognition. It's a disorder of behavior with a lot of disinhibition and personality change. A lot of symptoms that are often kind of misdiagnosed as bipolar syndrome. So it gets into that overlap with schizophrenia sometimes. I was very interested in it, and it also, there's a known role for tau in that disease. So that was kind of the molecular connection between the two for me as a lab focused mainly on tau. So we were working with a mouse model, a transgenic tau model. That was a new model that we hoped might be a good model for understanding frontotemporal dementia. And we got the mice, we grew them up, we tested them when they were about a year old, and they looked basically normal. And we were kind of disappointed. They didn't really have much of a phenotype. It wasn't promising that they were going to be a good model to do much with. But knowing that it's an age-related disease, we can't put them back on the shelf and say, well, let's check them when they're a little bit older, and see maybe something will develop in later life. And we started to get notices from the animal team taking care of the mice, changing the cages, that there were a lot of skin lesions and health checks. So so they're constantly having us come up and check on these mice because they have these lesions on them. And that's not uncommon in mice that happens to some extent. But this was just with this line, it was happening a lot. And when we started up there, it was not the typical kind of scratches that they would get with aging. It was always on their face, and they were just kind of had these raw spots on their face. And then we finally checked that, it wasn't every mouse in every cage. And we finally checked, and it was only, when we checked by genotype, it was only the mice that carried the tau transgene that had this. And their siblings in the cage who were the non-transgenic normal mice didn't get it. And that was kind of a light bulb moment where it's, okay, now maybe this is really something, this actually seems to be related to having the tau gene. But I think if I didn't have clinical training, I don't think I would have pursued it very much, because who wants to, follow up on a skin lesion in a model of a dementia? But the key is, one of the features of FTD is compulsive behavior and repetitive behavior. And some patients actually compulsively start scratching at their skin, and they get a lesion and they keep scratching it, and it becomes a wound. We had people who scratch their cornea just from rubbing their eye constantly. And that sort of clinical familiarity with the disease, is that could that be what's happening in the mice? And to make a long story just a little bit less long, that ended up being the case. These mice had a compulsive behavior of it's mouse grooming. So mice kind of, when they groom, they do this, and they were just grooming themselves so compulsively that it was creating lesions on their face. And we published a really nice paper. One of my MD PhD students, who's now a UCSF neurology resident, worked this all out, and actually we figured out what region of the brain was driving this dysfunction. We figured out that it was due to a insufficient amount of NMDA receptor activation. We were able to identify a potential pharmacological approach to boosting NMDA receptor function that actually reduced that repetitive grooming in the mice. And so that's probably the best example where really knowing the disease well helped me really design the experiments that we were going to do and pick the questions that we were going to answer. That's what, obviously, as a PI, that's probably your most important job, is deciding what questions you want to answer, because there's always a million of them. But that clinical experience helped us really zero on that one as a clinically interesting question, I think.
Forming a lab, it's a whole different thing, right?
Yeah. Forming a business is exactly the right analogy. When I first got into UAV, one of the senior members of the faculty, I remember at a faculty lunch, described it. It's a franchise operation. Right? They're basically, when you get a lab, it's like you get a franchise. But it's your own business, and you've got a lot of control. And a lot of stuff you're not really trained to do, because it's not, you get selected for this job based on the ability to do research and publish papers. And there's a whole lot of other stuff that you have to do as a faculty member to be successful. One of the first things that I did was, and this was advice that came out of, a few of the, like the Howard Hughes book on how to run a lab, and a few of the kind of how to be a faculty member courses that I'd been in at meetings and at Gladstone, was to think about the culture of the lab that I wanted to build, and the environment that I wanted to have. Labs are all different and driven to some extent by the personalities of everybody that's involved. But I created a mission statement for the lab, which I thought was really corny. And every time somebody told me that was important to do in these courses, I thought that was, who's ever going to look at that? I hate those things. But actually now I show that mission statement, and I don't think I've ever changed it from what I wrote in the first month that I was here. I show it to every single person that I interview, every rotation student and postdoc, because it just kind of it sets up kind of what we're looking for. And so the people who want that kind of thing then are attracted to come and join us, and it helps kind of foster it and keep it going. And the people who want a different kind of culture kind of probably don't come here, because they find the better match for themselves. But it helps us keep the culture, and maintain the culture that we want. I think that was one of the key things to getting the lab started.
And the balancing of that, we were talking about your clinical work and your lab work. Now we're adding another wrinkle, which is your lab management work, developing a funding source. I mean, you've layered on top of layers all of the things that you have to, all the balls that you have to juggle on a daily basis. How do you do that?
I don't know that there's any secret to it. I started the job, and I was like, am I going to be able to? You talked about confidence problems. I think every time you take another, the next step in your career, going from graduate student to postdoc, postdoc to faculty, assistant to associate to full professor, you never really feel fully prepared to make those leaps. And probably if you do wait until you really feel you're 100% ready, you've waited too long. You never really feel, I think, like you're ready to do that until you get in and start doing it, and you're like, and I was like, I don't know if I'm gonna be able to, my goal when I was a postdoc was to get a faculty job. My goal when I got a faculty job was, can I get an R01? My goal when I got an R01 is, can I get another one and get promoted to? So it was always just a challenge for myself. can I do it? I don't know, but I'll try. And I think having good mentors is really a key, because everybody who's been successful has had to figure all this out. And so, being able to tap into their expertise and the tricks that they've learned along the way. I was talking to one of my graduate students this week about, writing grants and all the grantsmanship tips that people pass along. It's most of them are because they've gotten burned on it somehow. Like, most things I tell people is because I wrote a grant once and I didn't say this, and that was why the reviewer didn't like it. And so now we always say this, and it's just a series of all these little cuts, and eventually you learn enough of them that you stop making those mistakes, and the grants start to get funded. But the extent that you can kind of learn some of those from other people without making them yourself, obviously you can progress a lot faster. So I've been fortunate to have great mentors, both as my lab mentors in graduate school and postdoc, and then great division directors and department chairs who both were good at doing this stuff, but were also willing to take the time to read my grants, to give me advice on hiring and all that kind of stuff. Taking advantage of those. I guess first of all, putting yourself in an environment where those people exist for you is key, and then taking advantage of them, of all the opportunities you have to pick those people's brains is really, really helpful.
When we talk about mentors, often we're focusing on younger scientists and mentors and helping them develop their career. But I think lifelong mentors are also critical. Agree?
Absolutely. Yeah. I mean, still, we try to do that even, with our faculty, our faculty, and I benefited as a junior faculty member from senior faculty, and now able to be on some junior faculty mentoring committees. But, even now, I recently promoted to full professor, and as you say, I'm directing a few centers, but I still there's still learning. I'm still learning lots of things and learning from the people above me. And, what kind of makes it fun is, we all kind of are driven by a desire to learn new things. That's kind of why we're in science. And that includes learning how to do new things. And so I think, you're never too old to have good mentors teaching you, how to take the next steps and how to keep, even if you think about kind of quote unquote staying the same, once you kind of level out as a full professor, there's not a whole lot more to go above that. But, you also realize that, if you're going to have a 30-year faculty career or more, you maybe have sort of 5 of that as an assistant and maybe 5 of that as an associate. And then that leaves like 20, most of it is as a full professor, right? But the world changes a lot in over that time. And it's not the same, even from the time you become a full professor to the time you retire. I mean, things are changing, technologies are evolving, the questions are changing. I mean, you can't just stay the same, and you've got to continually be adapting and moving things along. I remember my dad was a private practice radiologist, and when he retired, give some remarks at his retirement dinner. And as I was thinking that through, I realized that all of the things that he did in the course of his day-to-day life, for the last 10, 15 years of his career, reading MRI scans and ultrasounds and nuclear medicine scans, like none of that was even invented when he was a resident. So none of what he was doing at the end of his career was stuff he learned to do in his training. All of that stuff was stuff that he learned and adapted and learned in mid-career and adapted and built. And it was a great lesson for me about, the same thing is true for all of us in science, right? I mean, there's new techniques coming out. The bar for what it takes to get a paper published in terms of the number of different approaches that you need to use keeps getting higher. And so you still need that mentorship and growth to keep things fresh and keep things at the cutting edge.
Two of my favorite quotes, which don't really relate, but if you want to think about it, they sort of do. One is, "It ain't over till it's over." And Satchel Paige, the famous baseball player, said, somebody said, "How old are you, Satchel?" And he said, "Well, how old would you be if you didn't know how old you was?" And it kind of is in that mindset of ongoing, sustained curiosity, which doesn't, like Alzheimer's, have a start date and an end date. but it's critical to how you look at the world. So there's this juxtaposition of, on the one hand, yes, technology is changing, the world is changing. On the other, there are some basics. There are some coordinates, a compass that you can look towards. So you balance. Everything we're talking about here, in a way, is about balance. Quickly, you clinical work and your research in the lab, do you rebalance that periodically? Right now, how long do you spend seeing patients, and how long in the lab approximately?
So I guess the one change that the way that it has evolved over the time that I've been here is I've kind of added what would probably be a third thing on that list, which is, so I've done half a day a week in the clinic, plus 2 weeks a year of attending in the hospital. That hasn't changed since I started as a faculty member. Really, that hasn't changed since I graduated from residency. Even when I was a fellow, that's what I was doing, as I said before. That's the true clinical care piece of it. The one, and then the rest of it has basically was basic science through fellowship and when I started here. The third piece that I've kind of grown since I've been here over the last 7-8 years has been clinical research, the human subjects research, which I didn't do any of that. I was completely trained in graduate school and postdoc as a mouse and rat person. So just kind of got started in human subjects clinical research as I've been here, and that has been, that really started when a clinical trial came along. It was an industry-sponsored trial that they were looking for someone to lead here, and happened to be kind of the first trial that was supposedly aimed at tau, which is the protein that my lab was studying, and we didn't have another neurologist who had the time or effort to do it. So I thought, well, learn how to run a clinical trial. And so I did that, and it went really well. We ended up being able to recruit really well. Of course, the trial didn't work. No great surprise in Alzheimer's disease. There haven't been too many successful ones over the last 10 years. But I learned a lot from it, and that led to another trial, and led to a bigger interest in this as a way to kind of keep that bridge open. Some of it is for the lab. When we find something in the mice, we need human biospecimens to validate those findings, and we need brain tissue, we need CSF samples, and it's you can get those from collaborators and from other institutions, but at some point it just becomes helpful to have that infrastructure in-house, or and so we wanted to build that here at UAB, and so got more involved in building the clinical research programs to have that cohort of a clinical core of patients, and then all of the biospecimens and data that come from them to foster that. But it also, I think, just provides an outlet for, again, making sure that the things that we are doing in our rodent models don't get too far detached from the actual human disease. We don't want to end up going down rabbit holes of studying something in a mouse model that turns out to be an artifact of the mouse model that doesn't apply to human disease. And so doing that, doing the clinical research has been another way. Again, it's partly from a professional point of view. It's been a way to kind of add another dimension and keep things fresh as things have gone on. But it also, I think it does still fit into the whole overarching goal of trying to do research that helps lead to new treatments for disease by having all that stuff ongoing, kind of right here at our institution.
We're doing this interview on September 17th, and of course, COVID is in our mind. And you said something that really, really intrigued me. You said, most of what we know about Alzheimer's is based on research conducted outside the South. Now, you're at the University of Alabama, of course. And a particular problem is that there are very few African Americans who have been included in previous studies. Talk to us a little bit about that, and how are you addressing that issue?
Yeah, so that has become the theme and the focus of our Alzheimer's Disease Center here. So as we were kind of building this clinical research program in Alzheimer's disease, again, I think it's from a mentoring point of view, really critical to always have a question. This is probably the thing I spend the most of my time with my graduate students drilling into them. When you start, always have the question that you're asking at the forefront of your mind when you start to do a new line of experiment. So we didn't want to just, start doing clinical research and just be doing it without a clear question in mind. And as we sat down and looked at, well, what are the questions out there, and what are the questions, of course, that we in Alabama are well positioned to answer. Alzheimer's disease is more common in the Deep South. A lot of things that drive increased risk of Alzheimer's disease are more common in the Deep South. That includes being African American. It includes, we're in the middle of the stroke belt, the diabetes belt, the obesity belt. So a lot of kind of metabolic, cardiovascular risk factors that overlie this. There's also a lot of social differences, especially in the older cohort that's now vulnerable to Alzheimer's disease. The way that they grew up in the in the earlier part of the 20th century, was a whole different kind of ballgame in terms of their educational opportunities and poverty levels and things like that. So, and what is the role of all those things, and how do those interact with genetics and other medical comorbidities? So, this idea of Deep South disparities came to be our focus for the Alzheimer's Disease Center, and trying to understand the reason for the greater risk of Alzheimer's disease in this region. And of course, from a practical point of view, probably the most important, it's fairly easy to gather data on the diabetes part of it and the obesity part of it and the vascular part of it. But the racial part of it, looking at understanding why African Americans are at maybe double the risk of Alzheimer's disease. I mean, the only way we can answer that is with the participation of a good sized cohort of Blacks and African Americans, and that's been a big problem overall over the history of the last 20 years nationally in terms of Alzheimer's disease studies. The rates of African American participation have been really low. And being a center in Alabama, we really have no excuse to not help fix that problem, really. And so a lot of our outreach efforts are focused on the Black community in our metro area and statewide, and really trying to get better representation. We've had we're just getting started. This is a relatively new program, but we've had a lot of success, and I think it is certainly doable. It just takes effort and concentration and focus on addressing that issue. It takes a lot of involvement of a lot of different stakeholders in the process. But we've been able to recruit a cohort that's 40-45% African American. I think that's the kind of representation that we'll need to be able to answer questions about comparing whites and Blacks in terms of the risk and their risk factors.
Where do you think we are at this moment regarding Alzheimer's disease? If people see the signs or somehow the signs are recognized, where is that person on this spectrum in the year 2020 looking forward for them?
Well, I hope that we are on the verge of some big changes. We haven't had a new treatment for Alzheimer's disease since I was in training. That's really come on the market, and the ones that we do have, they're helpful. We use them. They make something of a difference, but they're not game changers, and we definitely need more. And I think the amount that we've learned about the disease since the last time a new drug came on the market to help treat it has been phenomenal. And I think we're on the verge. We may have one next year if aducanumab gets approved. We've had the first one submitted for approval over that almost 20-year period now. And so I don't know whether it'll get approved or not, but we may have a new drug next year. And if it's not that one, I think there'll be something. We're not going to go another period anywhere near that long. There will be some new things coming along. We have a lot more as a field. There are a lot more different kinds of irons in the fire between small molecules and antisense oligos and antibodies, and targeting not just amyloid but tau and many other things as well. And so I think it's an exciting time. And I think that, the treatment side and the diagnostic side can't ever get too far ahead of one another. Because if you can't treat a disease, that's what we can run these amyloid PET scans, but unless we have a treatment, the insurers won't pay for them because there's not a big impetus to have the precise diagnosis. And if you don't have a treatment, if you don't have a diagnosis, then you don't know who a new treatment would go to. So you have to have both. And the diagnostic side, we talked earlier about the blood tests for amyloid and tau that are coming along. I think we're making good progress on both sides of these, and I think it's only a matter of time, and I think a relatively short time before those really come into play and start making a difference in the clinic.
Since I focus on communication training for our Giannini Fellows, how would you rate the importance of communications and any quick insight?
Communication is important even when you're a postdoc, but once you get to be faculty and the actual doing the experiments becomes less and less part of your job, communication becomes almost all that you do. Almost everything. Your real job as an academic is to write papers and to get grants to get the money to be able to write the paper. So it's largely writing, but then the speaking part of it is also that helps kind of build your reputation. In some cases, like in the Giannini interviews, that giving the public the talk is part of getting the grants that enable you to get the paper. So communication, and then of course the teaching component is an important part of our job at a university setting. So it's absolutely fundamental to what you do. And I really think since the written is so critical, the papers and the grants are really the fundamental currency in academics. Really thinking and good training in biomedical writing, because biomedical writing is a little bit different bird than creative writing, and what you kind of got a lot of the things that we all get taught in middle school and high school. We have to kind of undo a few of those things to do good biomedical writing. So I think that's probably one of the key things to focus on in terms of communication success.
Dr. Erik Roberson, 2004 AP Giannini Fellow. This has been absolutely wonderful, and thank you very, very much.
It's been a lot of fun. Thank you, Jerry. Enjoyed it.